thank you for the invitation. Thank you, Miriam, and congratulations for this excellent meeting. And today I will present some recent data about the evolution of tRNA synthetases and the implications to the organization of genetic code. And let me see here. Okay. Here you can see an overview of the translation process. In the first step here, we can see the amylosylation process. This is a very important process during the translation, in the translation system, because in this process, we attach an amino acid in an tRNAs, and this process is very, very specific. And after this tRNA goes to the ribosome and start the protein synthesis. I will focus my talk today in this first process here, and basically the aminoacylation, and more specifically in the evolution of this group of enzymes that is the aminoacyl tRNA synthetases. And the relation between tRNAs and the aminoacyl tRNA synthetases established what you know as the genetic code. And we listen a lot about this organization. This is a very important code in biology. And the specificity, and not only specific, but the high fidelity in the biological system is due the correct interaction between the aminoacyl tRNA synthetases and the amino acid and the tRNA and the cognate tRNAs. So I will talk about the two main elements in this process. The first one is the aminoacyl tRNA synthetases. How can you see? And the aminoacyl tRNA synthetases participate of the many processes in the cell. But here I will focus on only in the protein synthesis, and more specific only in the process of aminoacylation. In general, we have the same reaction. We can find two classes of aminoacyl tRNA synthetase, class 1 and class 2, as you can see here. And each class have 10 amino acids. And we can see a symmetry in this point. And in general, class 1 and class 2 do the same reaction. Evidently, we have some details that is different, but in the process as well, we have the same reaction, and that you can see here. And the first step is the activation of amino acid. In this step, an ATP and an amino acid bind in the catalytic site of the cognated aminoacyl tRNA synthetases. And in this moment occur what you say activation of the amino acid. In this process, the ATP is broken and bind in the amino acids. After this, tRNA cognate bind in the aminoacyl tRNA synthetases, and these amino acids transfer to the CCA of the tRNAs. In the tRNA now attached on amino acids is released to participate of the protein synthesis. And a very interesting point here is when we analyze the way of the interaction between the tRNAs and the aminoacyl tRNA synthetases, we can see that you have asymmetry in between the class 1 and class 2. Both classes have 10 amino acids. And a very interesting point here is that the class 1 bind in a side of the tRNA, and class 2 bind by the other side of the tRNA. This characteristic suggests that during the evolutionary process, we had coevolution between class 1 and class 2. The other important element in this process is the transfer RNA. The transfer RNA is a very interesting molecule. It's a short molecule, have around 75 nucleotides. And a very interesting point here is that you have many different tRNAs, but all of them have the same secondary structure and the same tertiary structure. And other very important thing here is when you look to the gene structure in the genome to the tRNA gene, we have in all cases the 5' half that we can found the anticodon and the 3 half when we can found the acceptor arm. This part are codified, it's not codified, but are separated by an intron, or in other situation by multiple introns, or in some cases these two parts are in split genes. This is a very interesting characteristic because it's like we had two entities that work together to do mature tRNAs. So the first question is, how was the connection between amino acid tRNA synthetase and tRNA established? Here I will also show this picture. Romeo showed a similar picture. And it's a very interesting characteristic is that is when we look for the hydropath of the dinucleotides in the anticodon and the amino acid, we can establish a connection and hydropathic correlation. For example, if you have an amino acid very hydrophilic, when you look for the dinucleotide in the anticodon, the dinucleotide is also very hydrophilic. Well, this is a very interesting because when you look for the structure of the tRNA, the amino acid is attached in this portion here, in the CCA, in the acceptor arm, and the anticodon are here. And the anticodon and the acceptor arm is separated by 75 angstrom. And when you look for the points of interaction in the amino acid tRNA synthetase, it's a totally different points of interactions. And in 2015, I published this paper about the revolutionary history of the tRNAs, and the data, the results of this work suggest that the tRNAs evolved by changes in the second base of the anticodon. And in 2014, we published this other paper here. And when you look for this is a branch of the tRNA tree. And you can see here, alanine tRNA is class 1. When we change the second base of the alanine, will change to the aspartyl. And when you change the second base, we change the class of the amino acid tRNA synthetases. And we also change of the hydropathy of the amino acid and the anticodon. For example, you can see here, alanine hydropathy is 0.2, and the anticodon is 0.4. When you change the second base in these tRNAs, we had an hydropathy of the amino acid 0.9, and the base and the nucleotide also 0.9. This is a very interesting point because it sounds like a coevolution between the tRNA diversification and the diversification of amino acid tRNAs, class 1 and class 2. So we have two other questions. Who came first, the acceptor arm or the anticodon loops? And the other question is how to explain the correlation of hydropathy between amino acids and anticodon. And this time we need to remind that you have a point of attachment of the amino acid here and the anticodon are here. So far. And in the literature, we can find two main hypotheses about the first part of the tRNA and how was the first code. And we can find a little papers about the operational code. And the operational code is based in some experiments when the researchers separated only the acceptor arm, and it's only with this part of the tRNA is possible amino acylated by the amino acid tRNA synthetases. And from this data, it suggests that the first part of the tRNA was the acceptor arm. But there are a very interesting paper by Shimizu in 1995, when Shimizu did a very interesting experiment, and he used only the anticodon part of the tRNA, and only with this part of the tRNA what's possible amino acylated the tRNA, in this case, without the presence of the amino acid tRNA synthetases, only in the presence of dipeptides. So, and we have data from the capacity of the anticodon loop and acceptor arm to bind specifically amino acids. To try answer these questions about what was the first part and how was established the connection, the hydropathic connection between the amino acids and the anticodon, we did these two works. And this first work, we analyzed only the Glycial tRNA synthetases. This is a very interesting synthetase class 2. And more recently, Pedro did an excellent work and analyzed the natural history of class 1. And now I will show you some data that try understand better how was established these connections. So the strategy that we used was the reconstructed the ancestral sequences. This is a very interesting strategy when you are talk about very ancient molecules. And basically, the methodology is we took in the database a modern sequence for the protein that I want to analyze. And from this modern pool of protein, I reconstructed on a phylogenetic tree. If from this phylogenetic tree, I reconstructed the diplet node. And the diplet node is the more ancient protein for your tree. Okay, so this way we have constructed the ancestral sequence for glycine, Glycial tRNA synthetase, for two structures and complete deletion. This is more, we have more constraint. In the sequence in general is short. And we used other strategy that is partial deletion, when you try look for the and secondary stage in the evolution of this molecule. So in A and B, you can see the ancestral structure by the complete deletion, and in C and D by the partial deletion. So the first analysis that we did was test the capacity of this molecule binding specifically ATP and the amino acid, in this case, glycine. And you can see here is that the ancestral amino acid tRNA synthetases and bind in red ATP and in blue glycine. And when you look for the other ancestral, they also have the capacity to bind the ATP and the glycine. This is very interesting because when you look for this more ancient part here in A and in B, this ancient part is the catalytic site of the amino acid tRNA synthetases. And the other analysis that we did was, we did an experiment of molecular docking. And this experiment, we put the tRNAs and we put the ancestral enzyme, and you test if it's possible an interaction between the tRNA and the ancestral amino acid tRNA synthetases. In this case, we test if the anticodon loop had capacity to bind in the ancestral tRNA synthetases. And remember, this part of the amino acid tRNA synthetase is the catalytic site. In the modern protein, the anticodon doesn't interact with this part of the protein. But when you look for the ancestral sequence or the ancestral structure, we can see that it's possible an interaction between the tRNA loop and the anticodon catalytic site of the ancient protein. And a very interesting point here is this interaction is in the bases, in the second base of the anticodon. As you can see here, the interaction. When you put all compounds together, we can see that this picture in A, we have here the ATP in red, the glycine in blue. In green, you had the ancestral glycyl tRNA synthetases, and in orange, you have the anticodon loop of the tRNA. And this configuration make possible that this amino acid here could be transferred for this extremity here, like occur in the acceptor arm. So the first result here is, okay, it's possible that the ancient protein amino acylated the anticodon loop and in the orange of the biological system. And other very interesting point here is when the amino acid tRNA synthetases grow up and protected the catalytic site, the tRNA change your point of interaction. And in this moment, the tRNA don't interact more with the catalytic site, like we can see in the modern protein. And when you put the modern amino acid glycyl tRNA synthetases with the, we did structural alignment between the modern glycyl tRNA synthetase and the ancestral glycyl tRNA synthetases, we can see that during the evolutionary process, the tRNAs, the anticodon tRNAs that initially bind here, they change and rotate your interaction with this molecule. And this rotation here make possible that the acceptor arm interacted directly with the catalytic site now. Okay. So from this data, we suggested how was the evolution of the Glycial tRNA synthetases, and in our model, we had a variant part, and this variant part was the catalytic site, and this catalytic site had capacity to bind in the anticodon loop, and also had capacity to bind in the ATP and the amino acid, in this case, glycine. When the protein grow up, the tRNA change your point of interaction and move out of the catalytic site and interactive as you know in the modern protein. In this point here, the anticodon domain of the amino acid tRNA synthetase is the last part to be incorporated in the evolution of the amino acid tRNA synthetases. Okay, so this is very interesting because now you can have an idea why the anticodon and the amino acid had the similar hydropath. Because in the beginning of the interaction, both the anticodon and the amino acid bind in the same point of the protein. But we look for the class 1 if they had the same behavior. And here you can see, and we used the same strategy. We reconstructed the ancestral structure for each one and protein of the class 1. You can see here, and arginine, cysteine, glutamine, and here the last value. But here we did a little different analysis. In this case here, we test the capacity of the anticodon loop binding in the ancestral structure, but we also test the capacity of the acceptor arm binding in the ancestral structure. And for our surprise, when we look for the results, and we can see that when you have the ancestral structure, the anticodon, you can see here in black, the anticodon loop, and the acceptor arm in yellow. The anticodon loop, the acceptor arm binding in the similar point in the ancestral amino acid tRNA synthetases. And other very important here result that I need to highlight here is if you look for the point of interactions, is in general point unstructured. So this result suggests for us that initially the acceptor arm and the anticodon loop binding in the same point, and it's very important to say that here is the catalytic site of the protein, and in the modern protein, only the acceptor arm binding this point, okay? But the other important result here is this result suggests that the initial function of the interaction between the ancestral amino acid tRNA synthetases and the anticodon loop and the acceptor arm, it's not to codify or not to bind in amino acid. It was just to stabilize the structure. I think that was to stabilize both the tRNAs or the RNAs and sequence and the proteins that are in that moment a little unstable. When you look for the energy of interaction, here you have the interaction of the anticodon loop, and here you have the amino acid tRNA synthetase, and here you have the interaction of the acceptor arm, and the interaction, the energy of interaction is practically the same. This meaning that the affinity between the anticodon loop and the acceptor arm with the ancestral amino acid tRNA synthetase was the same. Okay, this again, with this data, I can think how was established the hydropathic connection between the anticodon and the amino acid, because in the early biological system, these two different things interacted within the same point of the enzyme, in this case, in the catalytic site. Okay, so we can see here an evolutionary pathways to the evolution of the amino acid tRNA synthetase class 1. And at the beginning, you have here arginine tRNA synthetase, and the beginning, and the anticodon loop, the acceptor arm bind the similar point. That point is the catalytic site. And when the protein grow up, one part of this tRNA move out of this part. In this case, is the anticodon loop. But the acceptor arm keep the interaction with the catalytic site. Okay, we can see this for arginine and also for the other proteins of the class 1. So from this data, I will suggest some ideas. The transfer RNA and amino acid tRNA synthetases are very old molecules in the biological system. I think that is a consensus in the literature. And the catalytic site of the amino acid tRNA synthetase is the oldest part. The data really indicated this. The acceptor arm and the anticodon loop at the origin of the biological system were able to bind at the catalytic site of amino acid tRNA synthetases. And here we suggested that the binding of the acceptor arm and the anticodon loop on amino acid tRNA synthetases initially worked to increase the stability. And with the maturation of the protein, we had a modification of this type of interaction. And the operational code and the anti-code co-evolved with the complexification of amino acid tRNA synthetases. And with the complexification of amino acid tRNA synthetase, the acceptor arm and the anticodon loop modified the bind sites and assumed different roles. At this time, the tRNAs assumed the modern structure. And the correlation of hydropathy between anticodon and its cognated amino acid occurred because at its origin, the anticodon loop bound at the catalytic site. This is a memory, the evolutionary memory of the origin of the biological system. I would like to thank many collaborators, Marco Rosset, Thais Gaudêncio, Francisco Projot, and that Marco and Francisco are here, Gustavo Capone, Sohan também also are here, and Pedro Henrique, I think Pedro are here also. Thank you.